Hey, so in this video, we're going to talk about the phototransduction. Now, phototransduction is how do the rods and the cones take those wavelength packages of energy that we call photons and turn them into electrical signal in both low light and bright light conditions, grayscale and full color vision. So let's start here. Now, when I look at photoreceptors, I'm talking about the rods and cones, and they really are modified neurons, and they kind of look like an upside-down epithelial cells. And when we start looking at the components, we have a cell body, we have synaptic terminals, and then we have two segments. An outer segment. Now, the outer segment is where the light arrives, and it contains visual pigments, photopigments. And these photopigments will change shape as they absorb light. So they'll be able to change from a bent form to a straight form. We'll come and see that in a few seconds. We'll get to there. So we have an outer segment, and then we have an inner segment that joins onto the cell body. Now, let's go through some of the terms, and then we'll see an image. The inner segment's connected by a cilium to the outer segment and to the cell body by an outer fiber. And the cell body is connected to the synaptic vessel by inner fibers, and also we have parts of the plasma membrane of the outer segment that form these discs. They fold back on themselves, and this is where the photopigments are embedded within the discs. Now, before we go into the diagram, let's say a few things about photoreceptors. They're very vulnerable to damage, and they degenerate in the retina. If the retina is detached, they will degenerate and they're lost. Also, intense light can damage them. And vision is maintained because the outer segment gets renewed about every day. So little tips of the outer segment will break off, they get phagocytized, and they get replaced because they're very sensitive to any type of damage. So let's have a look here. And let's start where we're at. So we are in the eye. This is the neural layer and the pigmented layer. So there's the pigmented layer. Remember, light would be coming in from here. So it's going to have to pass through the ganglion cells, through the bipolar cells, and then here to the rods and the cones. Now you can see why the rods are called rods. They're rod-shaped, cones, cone-shaped. And the other thing you can see here is that cones have almost a 1-to-1 ratio of cone to bipolar cell to there. So that, now let's compare this to the cones. That was the cones, to the rods. The rods, look at this, they converge information. So if this ganglion cell fires, it doesn't know if the signal came from here or here or here or all of them. So that's why when you use rods, your vision is not quite as clear. You don't have the same acuity. Whereas with cones, you have this 1-to-1-to-1 relationship, which gives you much more acuity, and that's one of the powers of the cones. So let's look at the parts of these. We have outer segments. So there's an outer segment of a rod, outer segment of a cone. Then we have the inner segments we can see here. And we can see that they're connected by a little connecting cilia. And then from here, the inner segment, we head towards the cell body, and this is the outer fiber, connects the inner segment to the cell body, and then the inner fibers head off towards the bipolar cells down here. And we can see that theme carries on with both of these. So rods are really sensitive to light, making them really perfect for night vision and peripheral vision. They contain only one type of pigment, where the cones contain a variety, 3 different pigments. So in the cones, each of the 3 pigments is really sensitive to different wavelengths, and we'll come to that. But in the world of rods, there's only one pigment. So rods, very sensitive to light, makes them great for night vision, peripheral vision, and they contain a single pigment. So vision is perceived in gray tones only. And as we said earlier, the pathways converge which can cause sort of the fuzziness. You lose some of that visual acuity, the sharpness of the image. And in some cases, you may have 100 rods converge on one ganglion cell. So that's a big receptive field, and you're never sure what part of that receptive field the signal is originated from when you fire that ganglion. So you're going to end up with some fuzziness. But hey, in low light conditions, it works really well. So let's go to cones. So cones have low sensitivity and they require bright light for activation. They react more quickly. They have one of three pigments, and we'll talk about those shortly, which allows for really colored vision. Okay? It's non-converging pathways, so that's going to give me detailed, high-resolution vision. And many cones have their own ganglia. So one cone goes to one bipolar cell to one ganglia, so you can really pinpoint where that image or that part of the image is in your visual field. We'll skip over the color blindness because I want to go here and talk about the visual pigments. So we're dealing with retinol, and the retinol is a really important molecule because it absorbs light. Now the retinol, it combines with one of four proteins, the opsins, okay, to form what we're going to call the visual pigments. By the way, the basis of this is vitamin A. So there's four opsins. Now, rods only have one variant, and this is rhodopsin. I always remember R, right? So I remember rhodopsin is in rods. And so rods only have one pigment, whereas cones have three. And each one of these absorbs in a different part of the spectrum, the light spectrum. We have those, the subcategories are the green ones, the blue ones, and the red ones. So they absorb different parts of the visual spectrum. And if you go back to a previous video, we saw the spectrum and where each one of these absorbs within. So depending on the wavelength of the light they absorb. So the other thing to know about the cones, because there's three different ones, that they overlap with each other. So cone wavelengths do overlap. So same wavelength may trigger one or more cone, enabling us to see a variety of hues and colors. So this is why yellow light may stimulate red and green ones, but if more red ones are triggered, then I would interpret that as an orange. So between those three variants in the cones, I can form all the colors that I'm used to seeing. Now, the retinol has isomers, so different 3D forms. Retinol's in the bent form in the dark, but when pigment absorbs the light, it straightens out. So retinol can change shape from bent to straight, okay? The bent form is called 11-cis retinol. The straight form is all trans retinol. Now the conversion of bent to straight is going to initiate reactions that are going to lead to electrical impulses along the optic nerve. And here we can see this is an example of rhodopsin. It's a visual pigment in rods. It's embedded in the membrane and it forms these discs, and inside I can have the retinol. You can see the retinol interacting with the rhodopsin, which gives me the visual pigment. And this is in the discs that I find within here. So let's talk about phototransduction, and that means I'm going to take a photon, a particle of light energy, and convert it into a graded receptor potential. So what happens is I have to capture the light. So the deep purple pigment of rods is rhodopsin. It's in the outer segment, and there's going to be 3 steps to its formation and breakdown, and then eventually regeneration. So I'm going to have pigment synthesis, pigment bleaching, and pigment regeneration. By the way, similar process in cones, just different types of opsins, and the cones require more intense light to do this job. So let's have a look. So pigment synthesis, so the opsins and the 11-cis retinol combine, I should say, to form the rhodopsin, and this happens in the dark. Okay. Pigment bleaching, when the rhodopsin absorbs light, the 11-cis isomer of the retinol, okay, changes to the all-trans form isomer. And retinol and opsin separate. So I'm going to break down the rhodopsin. And then regeneration, the all-trans retinol is converted back into the 11-cis isomer, and rhodopsin is regenerated. So I create it, it's bleached, I get a regeneration, and I'm changing the shape, right, the isomer shape as I go. And we can see it here, is that pigment synthesis, the 11-cis retinol derived from vitamin A combined with opsin to form rhodopsin, right? And remember, we're dealing with what's happening in the dark here. And then sorry about that, let's head back here. And then what happens is the pigment bleaching, light absorption by the rhodopsin triggers a series of steps in which retinol changes shape, okay? And 11-cis to the all-trans, and eventually it's released from the opsin. And then pigment regeneration, enzymes slowly convert the all-trans to its 11-cis form in cells, and of course it's going to require ATP to do that. So let's talk a bit about light transduction reactions. So light activated rhodopsin activates a G protein called transducin. So this is going to be a G protein system that we've seen in the past. And we're going to see transducin, remember transducin is the G protein, is going to activate something called PDE. Now this is an enzyme, and it's going to break down cyclic GMP. Okay? So it's going to break that down. So in the dark, cyclic GMP, what's it doing in the dark? It holds the cation channels in the outer segment open. We're going to see a diagram. We're going to walk through how this works, but let's get some of the basic players here. And sodium and calcium are going to enter and depolarize the cell. Now what happens in light? The cyclic GMP breaks down, the channels close, the cells hyperpolarize. Now it's kind of odd that hyperpolarization is a signal for vision. You would think you would polarize to get things going, like to fire a neuron eventually. But the secret here is this hyperpolarization is going to turn off in inhibition, and we'll come to that. So by hyperpolarizing, I'm going to stop one cell that's inhibiting another cell, and therefore that cell, if the inhibition's gone, will then fire. I know it's a little convoluted, but hang in there. So let's look at this second messenger system. So we have basically the first thing, light comes along, and we have the activation. We have the G protein, which is transducin. Okay, let's walk through it. So retinol absorbs the light, changes shape, visual pigment activates. Then what happens is visual pigment activates the G protein. The G protein, okay, transducin, activates an enzyme, and the enzyme in this case is PDE, and that stands for phosphodiesterase. So it's an enzyme, ends with A's. Now what's this enzyme do? Look at it. It seems familiar but different than the second messenger systems we've seen, because I'm not making cyclic AMP. What I'm going to do is I'm going to take cyclic GMP and turn it into GMP. And you're going, why? Well, if there's cyclic GMP around, it is keeping these channels open. And that means I can, sodium and calcium can travel back and forth. So let's have a look at that. So PDE converts cyclic GMP into GMP, causing a decrease in cyclic AMP. Those levels start to fall. Now what's that going to do? As the cyclic GMP levels fall, the gates, because cyclic GMP has been keeping gates open, so as their levels fall, the gates are going to start to close, and now what I'm going to do is I'm going to hyperpolarize this cell. All right, now little thing, let's head here. Photoreceptors, bipolar cells generate only graded potentials, EPSPs, IPSPs, not action potentials. So what happens is, if when light hyperpolarizes the photoreceptor cell, so I'm going to hyperpolarize rod or a cone, and when I do that, they're going to stop releasing, and here's the key thing, an inhibitory neurotransmitter glutamate. Now, so this rod in the cone, when not in use, is releasing glutamate. And remember, glutamate's an inhibitory, and it's telling the bipolar cell, do not fire. But then all of a sudden, when I get rid of the cyclic GMP, this cell now hyperpolarizes, and I do not release the inhibitory neurotransmitter, glutamate, which means the bipolar cell is no longer inhibited, and the bipolar cell can carry on, do what it wants to do. Bipolar cell no longer inhibited, depolarized, release a neurotransmitter onto the ganglion cells, and the ganglion cells can generate the action potential. All right? And you can walk through the stages that are in the book and in the notes. It walks through all those stages. And it's a really good step-by-step. And it walks through what happens in the light and what happens in the dark. But we have basically described that. All right. So I'm just going to talk a bit about light adaptation and dark, because rhodopsin is so sensitive that even in low light, it starts to basically bleach it. In bright light, the bleaching occurs very fast, that the rods become very non-functional. Now, the cones respond to bright light. So activation of rods and cones depends on what we call light activation or dark adaptation. Now, light adaptation, this is when you're moving from darkness into a bright light. So you're in a dark room, you head off in a beautiful warm summer day, a lot of brilliant sunlight, and you get that bit of a glare for a short period of time, because both the rods and the cones get stimulated quite quickly, and a large amount of pigment gets broken down and gives you that glare. Now, the one way you try and compensate is your pupils constrict. Now, give yourself 5 to 10 minutes and your visual acuity will improve, because the rod system turns off, the retinal sensitivity decreases, and cones and the neurons rapidly adapt to allow you to now see in the bright light. How about if I'm in bright light and I walk into a dark movie theater, right? I'm going to have trouble seeing everything at first. And the reason is that when I'm moving from bright light into darkness, we see blackness, because as soon as I get into the darkness, remember, cones need bright light. Now all of a sudden there's no bright light, cones stop functioning. But when I was out in the bright light, my rods had become non-functional. I had bleached the photochemical. So it's going to take a while for the rods, now I'm in the dark, to start to build up the photochemical. So bright light bleached the rod pigments, and so they're still turned off. The pupils are going to dilate to try and gather as much light. And the rhodopsin accumulates in the dark, so retinal sensitivity starts to increase. Transducin returns to the outer segment, and it takes maybe 20 to 30 minutes till you get full dark visual acuity, the best you can have, because it takes that time to regenerate everything. Okay, so that's a quick run through. Hopefully it was worth your time. Give a thumbs up, give a like. Feel free to subscribe to the channel, or drop a comment in the down below as to what you'd like to see covered, or if you want any other videos made. Thanks for your time, and we'll talk to you soon.